Are you really worried about biology exam 2026? Because you have only one day for your preparation before the exam? Don't worry. Start your preparation today. I am going to tell you very important questions from each chapter that is purely based on the blueprint, using which you can score happily 60 plus marks and improve your percentage. In future, I am going to tell you how to score 70 out of 70 also. Sexual reproduction in flowering plants, total marks expected 11, 1 mark, 2 mark, 3 mark and 5 mark. And these are the questions to be studied in priority. Human reproduction, total marks 10, 2 1 mark, no 2 mark, 1 3 mark, 1 5 mark. And here prioritize practicing male and female reproductive system diagram, sperm diagram and mammary gland diagram because you will get one of the diagram for 5 marks. Reproductive health, total 6 mark, 1 1 mark, 1 2 mark, 1 3 mark. For 2 mark, focus on amniocentesis, sterilization procedures in male and female, natural methods of contraception and MTP. For 3 mark, focus on IUD, types and examples, principles to prevent STDs, infertility and causes of infertility, and don't forget to read assisted reproductive technologies. Principles of inheritance and variation. I'll double tag this chapter because total 13 marks, 1 1 mark, 1 2 mark, no 3 mark, 2 5 marks. For 2 mark, focus on test cross and its significance, law of dominance, its definition, codominance with example, linkage and recombination, very important topic, linkage and recombination, sex determination, and very importantly, pedigree symbols. You will definitely get either linkage recombination or pedigree symbols. Since there are 2 5 marks question, I have categorized them into 2. One category, monohybrid cross, dihybrid cross, incomplete dominance. Among these, you will get 1. For second question, it will be in a hot, that is last main question. They may ask applied questions in terms of Mendelian or chromosomal disorders. Molecular basis of inheritance. This chapter also can be double tagged. Total 13 marks, 1 1 mark, 1 2 mark, no 3 mark, 2 5 mark. For 2 mark, focus on practicing transcription unit diagram, post-transcriptional modifications, splicing, tailing, capping, and types of RNA polymerases in eukaryotes, their division of labor, functions and all, replication fork diagram, transforming principle. In this chapter also there are 2 5 marks question. For direct question, focus on the concepts, features of DNA double helix, properties of human genome, steps of DNA fingerprinting and its application, Griffith transforming principle, Hershey and Chase experiment. For applied 5 marks question in last main, focus on topic, replication, transcription in prokaryotes, lac operon concept, translation and replication. Evolution, total 7 mark, 2 1 mark, 1 2 mark, 1 3 mark, no 5 mark. For 2 mark, focus on homologous analogous organs with examples in plants and animals, adoptive radiation with example, Hardy-Weinberg equilibrium and factors affecting it. For 3 mark, focus on Miller experiment diagram, stages of human evolution, order, industrial melanism, theories of evolution, Big Bang theory, abiogenesis and biogenesis. Human health and diseases, total 11 mark, 3 1 mark, 2 2 mark, 1 3 mark, 1 5 mark. In all categories, we could expect the question from diseases and their causative agents. For 2 mark, focus on infectious and non-infectious diseases with example, active and passive immunity difference, vaccination and immunization difference, allergy, antibody and chemical produced during allergy, chemicals used for the treatment of allergy, autoimmunity, definition and example. For 3 mark, focus on primary and secondary lymphoid organs, cancer, causes, treatment and diagnosis. For 5 mark, focus on topic, replication of HIV or retrovirus, innate barriers of immune system. Remember, the question from human health and disease can be asked in the last main, that is applied question. Microbes in human welfare, total 7 mark, 2 1 mark, 1 2 mark, 1 3 mark, no 5 mark. For 2 mark, focus on household products, distilled and non-distilled alcoholic beverages with example, microbial flocks, BOD and its significance, biogas plant diagram, symbiotic and free-living nitrogen-fixing bacteria. For 3 mark, I suggest you to only depend upon bioactive elements, their uses and source. Biotechnology principles and processes, total 7 mark, 2 1 mark, no 3 and 2 mark, 1 5 mark. I have categorized 5 marks question into 2 set. In one set, we could expect question from recombinant DNA technology, schematic representation based question, PBR 322, selection of recombinants, and this set of question can be expected for applied question. And for mixed question, that is tabular column type question that you find in question bank, they may include the concepts like electrophoresis, competent host isolation of DNA, PCR, and restriction endonucleases. Biotechnology and its applications, total 7 mark, 1 1 mark, no 2 and 3 mark, 1 5 mark. For 5 marks, focus on concepts, advantages of genetically modified organisms, uses of transgenic animals, gene therapy, RNA interference, and recombinant insulin production. Organisms and populations, total 6 mark, 1 1 mark, no 2 and 3 mark, 1 5 mark. For 5 marks, focus on the processes that affect the population growth, that is immigration, emigration, mortality, and natality. Growth models, exponential and logistic growth, their equation and all, and mainly focus on population interactions with examples. Ecosystem, total 4 mark, 1 1 mark, no 2 mark, 1 3 mark, no 5 mark. For 3 mark, focus on steps of decomposition. In particular, they act simultaneously, that is fragmentation, leaching, and catabolism. Food chain and food web differences, examples. Ecological pyramids, pyramid of number, biomass, inverted pyramid of biomass and energy. Among these, you may get one. Also, limitations of ecological pyramids, very important. Biodiversity and its conservation, total 4 mark, 2 1 mark, 1 2 mark, no 3 and 5 marks. For 2 mark, focus on types of biodiversity, species area relationship including formula, Eville quadrant of biodiversity loss, in situ and ex situ conservation, examples. Finally, all the very best for your exam. Get prepared well. After examination, don't forget to comment here how this video helped you to get a good marks.